Rhynia. Rhynia is a fossil plant. It is structurally simple but evolutionarily crucial early Devonian vascular plant. Okay, early Devonian vascular plant, and it is important while studying fossil because it is a direct fossil evidence for the origin of land adapted features such as vascular tissue, then cuticle, stomata. So basically, this you can say one of the most primitive vascular or the most primitive vascular plant, and it gives an idea about how evolution of vascular plants took place on planet Earth. So introduction for that, the fossils of the genus Rhynia were discovered by Robert Kidston and William Henry Lang during 1917 to 1921. They were obtained in the Rhynia chart. This Rhynia chart, this is basically a place in Scotland. It is Aberdeenshire in Scotland, and this fossil dates back to early Devonian period, about 410 million years ago. And the preservation of this fossil occurred through silica pre-mineralization. This mineralization part I have explained in my first presentation of fossils, and this mineralization was so proper that it retained the cellular and vascular details. When we will study the anatomy of it, we will be discussing about it. And as already said, it is regarded as one of the earliest known vascular land plants, and it is very crucial for understanding of the plant evolution. When we talk about the classification of Rhynia, of course it is kingdom Plantae, division Tracheophyta, and subdivision Rhiniophyta. You see a mark of this nature. This mark is given to extinct plants. So wherever you will get this mark, you remember that this is an extinct species or genus, or division, in this case subdivision. So class Rhiniopsida, order Rhyniales, family Rhyniaceae, and genus Rhynia. So let us first go to the external features of the plant, and according to Robert Kidston and William Henry Lang, Rhynia grew in these swampy areas, marsh areas, and these were associated with silica-rich hot spring environment, and this most probably could be the reason that silica mineralization had occurred. That means the soft tissues were replaced by silica, and the plant fossil remain intact, for which we are able to study the plant in the present day. And the name Rhynia has been given because of the place of discovery, Rhynie. It is in Scotland. Two species are identified, Rhynia goinivogeni and Rhynia major, and the name itself suggests the Rhynia major would be a little bigger plant, and goinivogeni is a smaller species of Rhynia. Both the species are small, herbaceous herbs, leafless, no leaves, and vascular plants, means they have xylem and phloem. Rhynia major it was comparatively bigger than Rhynia goinivogeni. The external morphology, just we look at the diagram. This diagram we can have a look. So there you can see the plant body is differentiated first into a rhizome, isn't it? A horizontal rhizome is present, which is a creeping rhizome, and from it you find that an erect structure, and which is dichotomy in nature. Branched aerial shoot is present. The aerial shoots, you don't find any leaves, no leaves are present, so they are leafless. And of course, since rhizome is present, rhizoids you will find, but there are no roots, and this kind of organization is a primitive organization. So true roots absent, instead rhizome is there and rhizoids are present. The upright shoots arises from the rhizome and shows repeated dichotomous branching, and at the terminal end you can find that there are sporangia present in certain dichotomous branching. So if we talk about Rhynia major, the aerial stems, they attain a height of about 50 centimeter and measure about 1.5 to 6 millimeter in diameter, whereas Rhynia goini, which is, as I said, was comparatively smaller, it reached a height of about 20 centimeter and about 1 to 3 millimeter in diameter. And in both the cases, as we have seen the diagram, aerial branches were cylindrical, naked, and leafless, and exhibited repeated dichotomous branching. Branches, if you see in the last slide, they are gradually tapering, isn't it? So from here, if you see, then it is gradually tapering at the tip. So branches gradually taper towards the apices. This is a primitive shoot organization. Okay, so this characteristics is a primitive shoot organization. And terminal ends, in some you find pear-shaped terminal sporangia. Next, we have to see the internal features. So external features are very simple. Let us go to the internal features, which are also very simple. So internal organization, here we find rhizome and aerial stem. So both in internal structure we have to see, and when we see it, we will see that nearly they are same anatomy. When we talk about the slender aerial branches of Rhynie, Goini, we find that the vascular tissues were absent, so indicating an incomplete differentiation of the conducting system. Okay, the general anatomy of the plant stem reflects a primitive vascular organization. So primitive vascular organization means you will find that here there will be xylem, and xylem would be surrounded by phloem, a protostele. No, this is a protostele type of arrangement. So this we will see in the next slide. So now let us see. If you see in the diagram, you will find that there is a single layer, isn't it? Single layer epidermis, and it is formed in the outermost single layered protective tissue of the aerial shoot. And this epidermis is this thick part of the epidermis. This is the cuticle part. This is the cuticle. This is the characteristics of normally any epidermis, indicating early adaptations to terrestrial conditions and prevention of water loss. The epidermal layer, in areas you will find that they are being interrupted, isn't it? These places are interrupted by stomata. The stomata are the small gaps which are present, which will facilitate gaseous exchange. And if you see the stoma is surrounded by guard cells, just like your leaves. When we study, the guard cells are present, and in between the guard cell, the small space is the stoma, comparable to those of the modern vascular plants. And the presence of cuticle and stomata demonstrates an advanced physiological adaptation for life, cuticle preventing water loss and stomata for gaseous exchange, also for excess water. If excess water is there, that can be lost through transpiration. So characteristics of normal vascular plants. So if we go in detail, then we see that each stoma is having two kidney-shaped guard cells, isn't it? Two kidney-shaped guard cells, and stomata, they are absent in the rhizome. In the lower part, there is no gaseous exchange, okay, because it is below, normally subterranean. And the rhizome, you find rhizoids are present on the surface, and basically that is for anchorage and absorption of water. Next, let us go to the cortex. When we see the cortex, we find that the major area, this is the cortex. So major area is the cortex, and the cortex we can again differentiate. You will find an outer cortex. This part is the outer cortex. Okay, this part is the outer cortex. Here a single cell layer it has been given, which we can call it also as hypodermis. So it is below the epidermis. It is below the epidermis, and hence can be called as hypodermis. And then an inner cortex. So this is inner cortex. So So that means you are finding that there is some kind of tissue differentiation. When we talk about the outer cortex, now I've already said this part is the outer cortex, which is the single cell layer, but we see that it could be 1 to 4 layers. In this diagram it is one layer, and the cells are polygonal and parenchymatous cells. Okay, uh, and in this case outer cortex, as you see that they are very compact, isn't it? The cells are compact, one attaching with the other, so no intercellular space, and it is mainly hypodermis function, which is what? Mechanical support and protection. Let us go to the inner cortex. This is the inner cortex part. So inner cortex, it is a broad zone of inner cortex. The inner cortex is composed of thin walled parenchymatous cells. Okay, cells are loosely arranged and you are seeing intercellular spaces. Lot of intercellular spaces are present here. So what it does, it facilitates storage and internal aeration. This intercellular spaces, it facilitates storage and internal aeration. These intercellular spaces communicate with the external atmosphere through stomata present in the epidermis. So gaseous exchange it takes place. And the structural organization indicates that inner cortex functions as the principal photosynthetic tissue of the plant. So you can say the inner cortex will contain chlorophyll for performing photosynthesis. Next, let us go to the stele. Stele, as I've already said, this central region is the stele, and this compact inner portion, that is the xylem, and then surrounding the xylem is the phloem part. So this is the xylem, and surrounding the xylem is the phloem. Okay, and this is the characteristics of any early vascular land plants, and Rhynia is one of them. And already said that the xylem occupies the central portion of the stele and surrounded by the phloem. So this is a protostelic type of organization. Here, very important, we have to remember, xylem consists exclusively of tracheids. So vessels are not present. So vessels are absent. No vessels. Only you have got tracheids. And these tracheids show annular and spiral secondary wall thickenings. These are all characteristics of primitive vascular plants. Okay. So vessels absent, already told. The phloem formed a narrow zone around the xylem, was composed of 4 to 5 layers of thin walled elongated cells. So phloem is surrounding the xylem, and of course phloem cells, here it is obliquely placed cells. Okay, and oblique placed cells is suggests a simple conducting tissue specialized for translocation of organic solids. If we move on to the reproductive structures of sporangia, the reproductive structure, the sporangia are, as we have seen in the first slide, that they are borne singly at the tips of some aerial branches. So basically they are terminal in position, and the shape is slightly cylindrical, club shaped, and the diameter is slightly greater. The diameter is slightly greater than that of the subtending branch apex. Okay, so here that the diameter, and here the diameter is slightly greater. The sporangia are erect, terminal, and these characters are primitive reproductive connections. If you see the sporangia of Rhynia major and Rhynia goini-bogani, there are little differences as we have tabled out here. Here length 12 meter, but goini-bogani is smaller, 4 millimeter. 12 millimeter, 1.4 millimeter, and breadth here 1.4 millimeter, nearly same. Spore diameter is equivalent to 65 micron, here 40 micron. So goini-bogani, the plant is also small, as well as the sporangia structure is also small. Now when we talk about the sporangia anatomy, you will find that 3 layers are there: outer layer, middle layer, and an inner layer. The outer layer, middle layer, and an inner layer. Okay, 3 layers. Outer layer, it is epidermal, which will be thick and heavily cutinized, basically protection. Middle layer, it is 3 cells thick protective layer, and only the inner layer is delicate, enclosing the sporangial tissue. So this is the inner layer, which is delicate, and it has the sporogenous tissue. This wall differentiation provides mechanical protection and reduces desiccation. Desiccation means breaking down for the release of the spores. So they will break down, of course, but only after attaining maturity. And this sporangium, if you see the inside, you will find that the spores are homosporous. Homosporous means you can't differentiate between male and female, or the spores are all similar in shape and size, similar in size and structure, indicating homospory. And homospory is a primitive reproductive feature, and in advanced feature we find heterospory or heterosporous spore, where you will find that there is some differentiation of the spores. Now when we talk about the views on the gametophyte and sporophytic relationship, there are different views. See, now Pant, the scientist, in 1960, he suggested that Rhynia goini is representing a gametophytic phase of Rhynia major. That means, according to Pant, these two plants are not different. Goini is representing the gametophyte. It is representing the gametophyte, which is smaller in nature. This is smaller. And R major is representing the sporophyte. And it is bigger in size, isn't it? It is dominant. This is what, according to Pant. But Puri 1961 proposed the structures interpreted as sporophytes may actually be gametophytes. He just reversed what Pant said. So all these things just simply I am saying because these plants were reconstructed, and there are different views by different scientists in different working time, isn't it? Now let us go to the life cycle of Rhynia. When we talk about the life cycle of the Rhynia, so first you have to understand that this life cycle is a haplodiplontic life cycle. This life cycle is a haplodiplontic life cycle. That means it has got a haploid phase as well as a diploid phase. Here this is the sporophyte. This is twice N. This is sporophyte. Okay, this is the sporophyte, and this is the dominant phase. This is dominant. This is the dominant phase of the plant. Okay, so we've already discussed the mature sporophyte is bigger, it is leafless, dichotomously branched, all these things already described. And we find the terminal sporangia. At the tip we will find the terminal sporangia developing at the tip of some aerial branches, isn't it? Inside the sporangia we find the diploid sporogenous cells. Okay, inside sporangia we'll find the diploid sporogenous cells. These diploid sporogenous cells will undergo meiosis. So it will undergo meiosis. Okay, and what it will form? It will form the sporangium. So inside it will undergo meiosis, and then the spores will be formed. These spores will be haploid in nature, and the spores will then germinate. Okay, so it will multiply from 1 cell to 2 cell, 2 cell to 4 cell. Germination will be there, and then these germinating spores will form another plant which is little smaller. Okay, this smaller plant is the gametophyte. This is the gametophyte. This is N. So it was sporophyte twice N. Inside the sporangium, spores are being produced, which is by the process of meiosis, and these spores are germinating to form the gametophyte. This gametophyte is bisexual. That means it will bear both the male reproductive structure and the female reproductive structure. Okay, these are bisexual, so bearing both the male and the female. Male is antheridium. Male is the antheridium, male, and the female is the archegonium. Okay, so antheridium, within antheridium what will be there? There will be antheridia will be produced. Within antheridia will be produced, or we can say male gamete. This is the male gamete which is being produced. Okay, The male gametes on liberation, it will has to move through water, then it will go inside the archegonium, within which what is there? The egg is present. So archegonia contains a single egg. So Antheridia will swim in water, move to the archegonium and fuse it with the egg. Once it fuses with the egg, then what will be formed? We all know, then zygote will be formed. Zygote. So this zygote will then undergo multiplication, and then what will be formed? Embryo will be formed within the archegonium. This embryo which is formed within the archegonium will develop into a young sporophyte. Young sporophyte is again now twice-n, because once the zygote is formed, zygote will be what? Twice-n. So within the archegonium, that means within the gametophytic plant, within the gametophytic plant, after multiplication, it will form embryo. Embryo will develop into a young sporophyte which is twice-n, and then that will develop into a mature sporophyte. So this is what you call the life cycle. The young sporophyte becomes free living and independent, completing the life cycle. So this is all about Rhynia. We can say the first vascular plant which human beings studied as fossil. So thank you.